glycerol lipids for their membranes. They must synthesize glycerol lipids without a source of glycerol. So the aquaglyceroporins in the cell membrane of the red cell and the parasite allow glycerol to enter in the path the organism can grow and divide. Shown here is a peripheral blood cell, peripheral smear of blood. Here's a single parasite, and here's when it's grown and divided into 32 daughter cells. So in one cycle, 32 daughter cells, 2 cycles, 32 squared, that's 1,000, 4 cycles, 1,000,000, 6 cycles, 1,000,000,000. That's why we have these surges of fever and chills in the malaria-infected people who have horrific malarial sludge, levels of malarial sludge passing through their circulations, causing end-organ toxicity. The last aquaporin point I'd like to make is that the aquaglyceroporins 7 and 9 reside in fat and adipose tissues on the far left, where we store extra energy. When our progenitors faced starvation, those who could store energy in times of plenty were at an advantage. But in the settings of fasting and starvation, glycerol is released from the adipocytes through aquaglyceroporin 7, where it's taken up by the liver. It enters through aquaglyceroporin 9, where the glycerol is remanufactured into glucose, allowing the maintenance of blood glucose levels even into the late stages of starvation. This is work of Jen Carberry in the lab, who's now a professor at Duke. The aquaglyceroporins are also freely permeated by arsenic. This is probably why the protein emerged evolutionarily. In some parts of the world, of course, arsenic is present in the drinking water. In the United States, we have pretty good drinking water, at least we thought we did until the Flint, Michigan episode. But arsenic is largely not a problem. But in eastern India and Bangladesh, the surface water is often contaminated with Vibrio cholera. So tube wells are put in to draw in groundwater, which is contaminated with arsenic, causing 140 million cases of arsenic toxicity every year, with an epidemic of hepatocellular carcinoma. We don't think our basic science can lead to the reversal of that, but it does illustrate the importance of the protein in the defense against stress situations. Aquaporins are also present in the roots and tissues of plants. Shown here on the right is a wild-type Arabidopsis, a small mustard plant. And on the left is a Arabidopsis engineered by Ralph Kaldenhoff at the University of Würzburg, so that the rootlets will have only 20% of the aquaporin expression. But to maintain the same stem turgor and foliage, as you can see, the engineered plant simply sends out a redundancy of rootlets. Plants compete with each other for water during times of drought. So all this work was proceeding and life was wonderful. And then early in the morning in October of 2003, the phone rang, a pleasant voice. "This is an important telephone call from Stockholm for Professor Peter Agre. Are you Professor Agre?" And I answered, "I sure am." And they proceeded to inform me that I would share the Nobel Prize in Chemistry with Roderick McKinnon, also a medical doctor, studying potassium channels up at Rockefeller University. And they passed the phone around, and I got to talk to the members of the Nobel Committee. But I was thinking as this proceeded that I was really a pretty miserable chemistry student in high school. I didn't think it was a wise thing to share with them that I received a D in high school chemistry. Probably mostly youthful rebellion, because my father and I had a few antagonistic points. And then I was starting to think about my high school chemistry teacher, whether he would aspirate his morning cornflakes when he hears the news. And I then was told that there'd be a press conference in 10 minutes and I should get ready for my day. So I sprinted to the shower, and Mary took that time to call my mother back in Minnesota. My father had died 8 years earlier. And she awakened Ellen early in the morning to share the news. And Mother thought for a moment. She said, "Mary, tell Peter that's very nice, but don't let this go to his head. He still needs to do something important." And we had been working on the aquaporins in malaria and decided to make a complete switch to work on malaria. The thing with the Nobel Prize is you can never control the levels of celebration or the implications. Driving home from the laboratory that day, this was posted, and I'd just like to defend my reputation that I'm really not the Wells Liquor Store's best customer. But here we are on the stage, the children, Mary, and the medal. And I think the most important thing about the Nobel is it's a public relations effort for science. Because when you pay your taxes, as all of you do, you're supporting the National Institutes of Health, the National Science Foundation, and much important scientific activity. So in part, this recognizes that. But in my case, it also gave us an opportunity to go in a new direction. I decided to become a malaria fighter full-time. Now, in this late date, malaria still is prevalent in some parts of the world, mostly the developed world, in particular in sub-Saharan Africa. Things are getting better, but there's still about 500,000 deaths each year, and these are children, young children. And in addition to those that die, there are some that survive it or are scarred for life. So we have a research program in 3 sites, 2 in Zambia, southern Zambia, which has turned out quite well. In northern Zambia and in Congo, there are huge challenges. And in Zimbabwe, where things were improving for a while, and now they're on the decline again. So Zambia is a lovely country. It was formerly known as Northern Rhodesia. They have a good liberal democracy with pretty good public health infrastructure. And the victims of malaria, of course, are the children of rural Africa, like these little boys playing in the village where we have one of our field stations. So they're doing fine. They're being prevented from getting malaria, but this little boy from the countryside was brought in near death with cerebral malaria. The brain swelling that malaria causes in some cases led him to near death. His life was saved by Dr. Philip Tuma, shown on the right, a Johns Hopkins pediatrician who's dedicated his career to working in rural Africa. His life was saved, but you notice he has a disconjugate gaze upward. It's because he's got cortical brain damage. His visual cortex is destroyed. He is blind and will never regain sight. So in addition to the deaths, there are probably millions of children like this poor fellow who survive but are never fully recovered. So it's a huge problem and an important problem.